The Protein Machine by Cassidy McKenna. Eek! Is that a slither I hear? I really hope there are no snakes here in the dark. I would never know because I don't produce a protein that lets me see heat, like snakes do. If there's a snake, it will surely find me. I keep losing my friends to these silly snakes. I wonder if I figure out how to make the protein, if I can make it in me. Hey, is there a friendly snake out there that can teach me how to see heat? Oh yes, little bunny, I can help you be eaten. Help you learn how to see heat. All living things have cells, and in some of my cells, I can make TRPA1, a protein that lets me see heat. The production of this protein starts in the nucleus because my cells are eukaryotic, which means they have a nucleus that stores genetic information. The first part of protein making process is called transcription. DNA has different genes that code for proteins. The one we are looking at is TAC, GAC, CGT, CCA, ATG, ATC. And this is it inside of the nucleus. Here is the gene. It is identified and unzipped by the RNA polymerase. And this process creates RNA using free-floating nucleotide bases that are around the gene. RNA is complementary to one strand of the two-stranded DNA. The difference between them are the DNA has thymine as a base and RNA has uracil as a base. Also, RNA has ribose as a sugar, while DNA has deoxyribose. The RNA for our protein is AUG, CUG, GCA, GGU, UAC, UAG. Be the RNA polymerase and unzip the DNA. There are 3 different types of RNA: messenger, mRNA, transfer, tRNA, and ribosomal, rRNA. The mRNA was just created and is now leaving the nucleus and heading into the cytoplasm for the next step, translation. It's going right here. Okay. In translation, mRNA, which carries the code for DNA, goes into the small subunit of the ribosome, which is constructed partly by rRNA. Here's a small subunit. Then the tRNA, which carries amino acids and has a set of 3 nucleotide bases called anticodons, connects to the large subunit of the ribosome in position A, and one by one connects to the corresponding sets of 3 bases of mRNA, called codons. The anticodons for this protein are UAC, GAC, CGU, CCA, AUG, and AUC. The first tRNA connects to the start codon in mRNA, even if it isn't the first codon in the line. The start codon is AUG. This is the start codon, and you see it's connecting in site A, and this is the tRNA, and this is the mRNA. After the connection is made, the tRNA moves in position P, and the next corresponding tRNA moves in position A. Once the second tRNA is connected, the amino acids that they are carrying connect as well. The ribosome connects the amino acids with peptide bonds. So this is the first tRNA that went in, and now since it moved to position P, there's room for the second tRNA to move into position A. And this is the connection that is made between the amino acids. Every time a new tRNA connects, all of the tRNAs move down into the next section of the ribosome's large subunit. There is 3 sections, A, P, and E. Once the tRNA is in section E and another tRNA enters the ribosome, the tRNA in section E leaves to repeat the process of producing proteins, and the mRNA also leaves the ribosome, but instead of being reused like the tRNA, is broken down into the bases to be made into more mRNA. The process of protein creation ends when the tRNA's anticodon connects to the mRNA's end codon. So this is the end codon, this is the anticodon that connects to it, and it doesn't have an amino acid. So the last anticodon does not carry an amino acid. The end codon in this protein is AUG, right here. Then the finished protein is disconnected from the ribosome and folds so it makes a three-dimensional shape. Our finished protein is made from the amino acids methionine, leucine, allicine, glycine, and tyrosine. So you're gonna give me the genes so my cells can make the TRAP-01 protein, right? No, I'm gonna use my amazing heat sensing powers to eat you. The end.